What if some of the most fascinating organisms on Earth were literally underfoot, shaping our world in ways you can't even imagine? They're quiet, often hidden, but their impact is, well, it's absolutely profound. Today, we're taking a deep dive into the extraordinary world of Homobasidiomycetes. Now, this isn't just some obscure scientific name. This is a massive and incredibly diverse group of fungi. It includes everything from, say, the humble button mushroom on your dinner plate to the silent destroyers of ancient forests.
That's exactly right. For this deep dive, we're going to unpack a pretty dense scientific chapter from Introduction to Fungi by Webster and Weber. Our mission really is to cut through the scientific jargon and show you exactly why these fungi are so incredibly important.
Yeah.
Not just for science, but, in our everyday lives. We'll explore their unique structures, their intricate life cycles, and their often overlooked ecological roles. Think of this as your shortcut to truly understanding these hidden architects of our planet, especially if you're looking for that deeper understanding without needing a microscope right in front of you.
Okay, let's unpack this then. When early scientists first tried to make sense of the vast, sort of unseen kingdom of fungi, it was a real challenge, wasn't it? You couldn't just put them neatly into plant or animal categories.
Not at all.
So, historically, classification relied heavily on what you could see, the outward appearance of the mushroom itself. The 19th century Swedish mycologist Elias Fries, for instance, developed a system based almost entirely on how the hymenium, that's the spore-producing surface,
I
right?
need the spore factory.
How that was arranged on the hymenophore, which is the food body structure that holds it up.
Exactly. He was looking at the macroscopic features.
So when Fries looked at these fungi, what were the most obvious visual characteristics that helped him categorize them? What did he see?
Yeah, good question. He focused on a few key shapes for that spore-producing surface. You had the classic agaricoid type.
Your typical mushroom shape?
Right, your typical umbrella-shaped mushroom with those delicate blades or gills underneath. We call that lamellate. Then there's the poro type, where instead of gills, the underside is riddled with tiny holes, almost like a sponge. Think of those tough bracket fungi you see growing on tree trunks.
Ah, okay.
He also noted the hydnoid fungi, which have tiny teeth or spines hanging down like miniature icicles.
Oh, cool.
And then there were the simpler forms, like the resupinate fungi, which are just a flat crust sort of pressed against the underside of a log or branch. And finally, the gastroid types, like puffballs, where the spores are produced entirely internally. They aren't actively shot out into the air. He was really trying to create some order from all this visible diversity.
I see. So early mycologists were essentially kind of judging a book by its cover, so to speak.
Pretty much, yeah.
But it sounds like nature sometimes finds the same elegant solution to a problem, even with completely different blueprints. This classification based on shape seemed intuitive, but what was the major flaw that emerged later?
That's precisely it. The major flaw was something called convergent evolution.
Ah,
This means that very similar physical forms, like gills or pores, evolved independently in completely unrelated fungal groups. So, for instance, you see those sponge-like tubular hymenia in fleshy mushrooms like boletus.
oh, there's a seps.
Right, but also in the much tougher bracket, fungi like Trametes. Totally different texture, different group, same structure. Or those spiny toothed hymenia, you find them in a Homobasidiomycete like hymenium, but also in pseudohymenium, which belongs to a completely different major group of fungi, the Heterobasidiomycetes.
Wow.
So yeah, this really challenges the idea that outward appearance tells the whole evolutionary story, because very different fungi can end up looking remarkably similar. It's almost like nature's playing a trick on us.
So if gross morphology, or that outward appearance, isn't reliable for a true classification reflecting evolution, how do we classify them now? What changed things?
Well, modern classification has moved from the visible to the molecular. We now rely heavily on molecular sequence data, particularly from nuclear and mitochondrial ribosomal DNA genetics.
Right, the DNA evidence.
Exactly. This genetic evidence has completely reshaped our understanding. It's revealed that what we once thought was a single cohesive group based on appearance, the Homobasidiomycetes, is actually distributed amongst 8 distinct evolutionary branches or phylogenetic clades. This clearly shows that those external features, while helpful for, identifying something in the field, are often unreliable for building a truly natural classification system that reflects evolutionary relationships. It's a fundamental shift.
And it really drives home that idea of convergent evolution, doesn't it?
Absolutely.
It's truly mind-bending to think these complex structures like gills and pores evolved not just once, but multiple times, independently across different lineages.
It tells us that these forms must be incredibly successful evolutionary strategies, right?
Hmm.
Regardless of the underlying fungal family.
Yeah, and it's thought that the earliest Homobasidiomycetes likely had simple, flattened, or resupinate forms, and then those more complex structures evolved multiple times later on. Just incredible.
It truly is. So we've seen how appearances can be deceiving and why scientists had to look beyond the surface. Now let's go even deeper, right into the very architecture of these fascinating organisms. To truly understand how they're built, layer by layer, when you look at fungi, you immediately notice fundamental differences in their construction, even just their texture. Take the agarics, for example, your typical fleshy, umbrella-shaped mushrooms with a central stalk and gills underneath.
Like a button mushroom.
Yeah, these are quite different from polypores, which are often tough, leathery, corky, or even woody bracket fungi, usually with pores, and they're often attached laterally to their substrate, like a little shelf on a tree.
Okay.
These texture differences reflect completely different building principles at a microscopic level.
And that's where their microscopic architecture comes into play, right? Scientists like Corner revolutionized our understanding by dissecting basidiocarp tissues and revealing three main types of hyphae, those thread-like filaments that make up the fungal body. So what are these fundamental building blocks?
Well, first, you have generative hyphae. Think of these as the basic Lego bricks of the mushroom.
Okay.
They are the fundamental building blocks, capable of producing all the other cells and the spore-producing hymenium. They can be thin or thick-walled and may or may not have specialized connections called clamp connections. They're the versatile ones.
Got it. The primary threads.
Then there are skeletal hyphae. These are like the steel rebar providing strength. They're largely unbranched, very thick-walled, and provide a rigid framework. Think of them as the mushroom's strong internal scaffolding.
Makes sense.
And finally, binding hyphae. These are much branched, narrow, and also thick-walled. They weave themselves between the other hyphae, acting kind of like superglue, binding everything together tightly.
That's a great analogy. So generative hyphae are versatile, skeletal hyphae are the scaffolding, and binding hyphae are the internal glue. Together, they explain why some mushrooms are so delicate you can crumble them, while others are tough as wood.
Precisely.
And based on these hyphal types, we classify fungal fruit bodies using what's called the mephutic system. Can you explain that?
Sure. A monometic basidiocarp is made up only of those generative hyphae. Most of the fleshy agarics you might eat, like Lactarius and Russula, are monometic, and they're often quite brittle, right? That's because they often have special globose, thin-walled cells called spherocysts mixed in, which contribute to that crumbly texture.
Ah, okay. That explains why they break so easily.
Then there's the dimitic type, which contains generative hyphae plus either binding or skeletal hyphae. For instance, Laetiporus sulphureus, commonly known as chicken of the woods, is dimitic with binding hyphae, making it a bit tougher, chewier.
Right.
And finally, the trimitic system includes all three hyphal types: generative, skeletal, and binding. Tremesi's versicolor, the toki tail bracket fungus, is a perfect example.
Oh, yeah, those are tough.
Exactly. If you've ever tried to tear one apart, you'll know how remarkably tough and leathery it is. That illustrates its strong, complex, trimitic construction.
It makes perfect sense when you think of them as building materials. But how does a mushroom actually form from these microscopic threads? It all starts from a tiny hyphal knot, right? Just an aggregation of hyphae on the main underground body, the dikaryotic mycelium.
Yep, from that little knot, the magic begins.
So what's involved?
Well, part of that magic involves special proteins called hydrophobins. These are secreted onto the surface of the hyphae, and they make the fungal surfaces non-wettable, or hydrophobic.
Like waterproofing.
Essentially, yes. This property is crucial because it creates air-filled channels within the developing fruit body. This allows for efficient gas exchange, even when conditions are very wet outside. It can even help protect the fungus from certain parasites or just getting waterlogged.
Clever stuff. It's all about protection in those early vulnerable stages, isn't it?
Absolutely.
Many agarics especially have these fascinating veils that protect the developing fruit bodies. Tell us about those.
Yeah, they're quite ingenious structures. There's the universal veil, which completely envelops the entire young mushroom like a protective little egg.
Okay.
As the cap expands, this veil tears. It often leaves a cup-like structure, the vulva, at the base of the stem, and frequently scales or patches on the cap surface.
Mm-hmm.
Think of a classic Amanita mushroom. That's how those distinctive spots and the basal cup form.
Right, the iconic fly agaric look.
Exactly. Then there's the partial veil, which stretches just from the cap edge to the stem, specifically protecting the young gills. It can be really thin and cobweb-like. We call that a cortina, seen in fungi like Cortinarius. Or it can be firmer, persisting as a ring or annulus on the stem, like you see on a common button mushroom from the store.
And based on the presence and development of these veils, we describe different developmental patterns, right? What are those?
That's right. Gymnocarpic development means the hymenophore, that spore-producing surface, is naked from the very beginning. It's never enclosed. You see this in genera like Cantharellus, the chanterelle, or Boletus.
Exposed early on.
Yes. In contrast, angiocarpic development means the hymenophore is enclosed during at least part of its development. This enclosure can be primary, differentiating beneath the primordium surface, or secondary, where hyphae actually grow out and around to enclose an already differentiated hymenophore.
Fascinating. Okay, speaking of gills, or lamellae, the details of their structure are truly amazing. Most gill-bearing fungi have gills that are wedge-shaped in cross-section, don't they?
Yes, that's the equihymenial type. The hymenium develops uniformly over that wedge shape.
And why that shape?
Well, it's actually an amazing adaptation to minimize spore wastage if the fruit body happens to tilt slightly off vertical. Keeps the spores falling straight down between the gills. And the fungus even has this incredible ability called gravitropism. It senses gravity and actively reorients its stalk, or stipe, to keep those gills perfectly vertical for efficient spore dispersal.
That's incredible. But then you have a completely different strategy with the inequihymenial gills, characteristic of the ink caps like Coprinus.
Ah, yes, the ink caps. Their gills are parallel-sided, and the basidia, the spore cells, ripen in zones moving up the gill. This leads to autolysis, or self-digestion.
They digest themselves.
Yeah, the gill basically melts into that inky black liquid as the spores are discharged. That's why they look like they're dripping ink. It's a rapid dispersal mechanism.
Wild. And you can even describe how gills attach to the stem, can't you? Things like free or decurrent.
Absolutely. Mycologists use terms like free if the gills don't touch the stipe at all, adnate if the entire base is attached, adnext for partial attachment, sinuate if there's an S-shaped curve near the stipe, and decurrent if the gills run down the stipe. All useful for identification.
Okay. And what's supporting these gills or pores?
Supporting all these intricate hymenophore structures, whether they're gills, pores, or spines, is the hymenophoral trauma. This is the central core of hyphae, running through the gill, pore, or spine, acting as its internal support structure. There are different types, like the divergent trauma seen in Boletus, where the hyphae spread out from the center, or the intermixed trauma with those globose spherocysts found in Russula and Lactarius, contributing to their brittleness.
And finally, we arrive at the hymenium itself. This is that palisade-like layer where all the action happens. It's composed of the basidia, which are the spore-producing cells, and often other sterile structures like cystidia and periphyses. What are those doing there? They sound like extras.
Cystidia are particularly interesting. They are varied, often in large cells that can pop up in different places within the fruit body, sometimes protruding quite prominently from the hymenium. Their functions are still being fully understood, honestly. But in Coprinus, for instance, they seem to act as tension elements, like little struts, physically helping to space and straighten the gills as they expand. In another species, Vulvariella bomicina, they appear to be secretory, maybe releasing water vapor, perhaps helping manage humidity. But for many cystidia, their exact role remains a fascinating mystery.
And periphyses.
Periphyses, also seen in Coprinus, are thought to play an important role alongside the basidia in the overall expansion and structural integrity of the fruit body as it grows.
Right. From their hidden microscopic structures, we move to their, well, undeniable influence. How do these quiet architects of the planet truly shape our world, from the forests to our dinner plates? Let's talk about their real-world significance.
Okay. Their ecological roles are vast and absolutely crucial. As saprotrophs, they are the ultimate decomposers.
The recyclers?
Exactly. Think of them as the planet's recycling crew. They're fundamental to global carbon cycling, breaking down tough plant materials like cellulose and lignin in wood and leaf litter. They release locked-up nutrients that would otherwise remain unavailable for new growth. Essential work. When it comes to wood degradation specifically, they have two main strategies, and they are strikingly different. First, you have brown rot fungi. A famous example is Serpula lacrymans, the notorious dry rot fungus, which causes immense economic damage to timber and houses.
Oh yeah, dry rot is bad news.
Very bad news. These fungi primarily destroy cellulose, leaving behind the lignin, which turns brown and cracks into distinctive cubes. It's still a bit of a mystery how they efficiently get past the lignin shield, but they produce oxalic acid, which lowers the pH, and possibly even small, highly reactive molecules like hydroxyl radicals that can kind of chew through it. This fungus, Serpula, has even been transported globally on old wooden ships because it's so adaptable to human habitats.
Wow. Okay, so that's brown rot. What's the other strategy?
The other is white rot. Fungi like Tremades versicolor, turkey tail, or the well-studied Phanerococcate chrysosporium are capable of degrading both lignin and cellulose. They effectively bleach the wood in the process, leaving it white and stringy.
They eat everything.
Pretty much.
Yeah.
Lignin breakdown is incredibly complex, though. It involves powerful oxidative enzymes like lignin peroxidases, manganese peroxidases, and lactases. They often use small diffusible molecules as charge carriers to attack the large lignin polymer. What's fascinating is that this process is often co-metabolic, meaning they don't necessarily use the lignin for energy directly, but they break it down primarily to get access to the more easily degraded cellulose underneath.
That's clever. Remove the barrier to get the prize.
Precisely. And this ability is a huge area of research because of its potential applications.
So they're basically running a tiny, super efficient recycling plant under our noses. That's incredible. And beyond decomposition, what about their interactions with living organisms?
Another vital role is their mycorrhizal associations, especially ectomycorrhiza with trees. This is a critical symbiotic relationship.
Symbiotic, so both benefit.
Yes, hugely. The fungus forms a thick sheath called a mantle around the fine tree roots and a network of hyphae, the Hardick net, that grows between the root cells, but mostly stays outside them. The benefits are entirely reciprocal. The fungus acts like an extended root system, exploring the soil much more effectively than the tree roots alone. It transfers essential mineral nutrients, particularly phosphate, which is often limiting, and water to the plant.
And what does the fungus get?
In return, it receives carbohydrates, sugars, produced by the tree through photosynthesis. It can be up to 10% of the plant's total photosynthetic output.
Wow, that's a significant trade.
It is, and you can imagine an entire forest connected by these underground mycelial cords, allowing carbon transfer between plants, even from sunlit trees to shaded seedlings. Some colorless plants, like certain orchids, even plug into this network to steal nutrients, a strategy known as mycoheterotrophy.
A whole underground economy.
Exactly. And this mycorrhizal dependency is why many of our most prized wild edible mushrooms, like the sep, Boletus edulis, and the chanterelle, Cantharellus sebaris, are mycorrhizal and cannot yet be cultivated easily on a large scale. They truly need their tree partners. Some are very specific, like Suillus grevillei only grows with large trees, while others like Amanita muscaria can partner with multiple tree types.
Fascinating stuff. So, beyond their massive ecological feats, how do these fungi directly impact us as humans? Well, for starters, there's food.
Definitely food. Thousands of Homobasidiomycetes species are potentially edible, though only about 40 are actually cultivated globally on a commercial scale. The top cultivated species include Agaricus bispinus, that's your common button, cremini and portobello mushroom, as well as Lentinula edodes, shiitake, and Pleurotus satelli oyster mushrooms.
And production is growing.
Rapidly, especially in the Far East. For Agaricus bisporus, commercial cultivation is quite sophisticated now. It involves growing them on specially composted straw and manure, where heat-loving thermophilic microbes play a key role in preparing the substrate. A casing layer of peat or soil is then added.
Why the casing?
The casing layer is crucial. It holds moisture, provides structure, and interestingly, bacteria within that casing layer actually help induce the mushroom fruiting.
No way! Bacteria help grow mushrooms.
They do. It's a complex interaction. And oyster mushrooms, Pleurotus ostratus, are great because they can be grown on agricultural waste products like straw or sawdust. However, their massive spore release initially led to respiratory problems called mushroom worker's lung, which spurred the development of low spore or sporeless mutants for cultivation.
Always unintended consequences. What about other cultivated ones?
Volvariella volvatia, the paddy straw mushroom, is notable for having perhaps the shortest cultivation cycle of any fungus. You can get a crop in just 8 to 10 days.
Incredible speed. Okay, beyond food, there's immense medical and biotech potential, right? You mentioned the white rot fungi earlier.
Yes, their non-specific lignin-degrading enzymes are being extensively investigated for bioremediation. They can literally break down stubborn man-made pollutants like polycyclic aromatic hydrocarbons, PAHs from oil spills, for instance, organohalogens like certain pesticides, and even explosives like TNT.
Cleaning up our messes.
Potentially, yes. It's
Okay.
a huge area of research. Their oxidative enzymes are also used in industry for more environmentally friendly bleaching of wood pulp for paper production and even for decolorizing textile dyes.
And then, of course, the medicinal fungi.
Right. Ganoderma lucidum, or reishi, is widely cultivated, particularly in China, for compounds called polysaccharoptides. These are believed to enhance the immune system and are often used as a complementary therapy alongside conventional cancer treatments. Plus, fungi from the Mersmiaceae family produce natural fungicides called strobilurins. These compounds, or synthetic versions based on them, have been developed into major commercial fungicides used worldwide in agriculture. In nature, they likely act as a way for the fungus to capture resources and defend against competitors.
Amazing potential. But as with any powerful natural force, there are also significant dangers we need to talk about.
Absolutely. Poisonous fungi are a serious concern, and misidentification can be fatal. The most infamous is probably Amanita phalloides, the death cap.
Just the name sounds bad.
It is. It's deadly. Its primary toxins, particularly alpha-amanitin, attack liver cells by inhibiting a crucial enzyme needed for protein synthesis. Phalloidin also causes cell destruction. What's particularly treacherous are the delayed symptoms. Someone might eat it, feel sick with gastrointestinal issues after 6-12 hours, then actually feel better for a day or so.
Oh, a false recovery.
Exactly. It lures patients and sometimes doctors into a false sense of security. But during that time, the toxins are relentlessly destroying liver and kidney cells. Severe organ damage resumes with a vengeance, often leading to liver failure, kidney failure, and death if treatment like a liver transplant isn't possible. Always, always be 100% certain of identification, or don't eat wild mushrooms.
Sound advice. What about others? The fly agaric.
Amanita muscaria, the fly agaric, the iconic red one with white spots. This one is more hallucinogenic than typically deadly, though unpleasant side effects are common. Its compounds, ibotenic acid and muscimol, mimic neurotransmitters in the brain, causing altered perceptions, vivid dreams, muscle twitching, that kind of thing. It has a long history of use in traditional ceremonial practices in some cultures, like Siberia.
Okay. Any others people should know about?
Well, Coprinus atramentarius, the common ink cap, is interesting. It's actually edible and quite good,
Unless.
unless consumed with alcohol. It contains a compound called coprine. If you drink alcohol even up to a few days after eating this mushroom, the coprine blocks an enzyme involved in alcohol metabolism, causing symptoms very similar to the anti-alcoholism drug, disulfiram, flushing, nausea, palpitations, general misery.
Good to know.
Another deadly one to be aware of is Cortinarius oriolanus and related species. These contain a toxin called orlanine, which causes severe delayed kidney failure, sometimes weeks or even months after ingestion. The long delay makes diagnosis tricky.
Scary stuff. And magic mushrooms.
Psilocybe, SPP, or magic mushrooms, contain the hallucinogenic compounds psilocybin and psilocin. These affect serotonin systems in the brain, leading to altered states of consciousness. An interesting visual clue is that their flesh often turns blue when bruised due to the oxidation of psilocin. Research is ongoing into their potential therapeutic uses under controlled conditions, but recreational use carries risks.
So incredibly important to be aware of the dangers. Beyond human consumption, some are pathogenic fungi, causing widespread damage in nature, right?
Absolutely. Take Armillaria malea, the honey fungus. It's a major root pathogen of woody plants, causing root rot and group dying in forests and gardens. It spreads aggressively via white mycelial sheets under the bark and these tough, bootlace-like structures called rhizomorphs that can travel through soil.
Bootlaces of death for trees.
Pretty much. And it can survive for decades as mycelium within dead wood, waiting for a new host. Intriguingly, the mycelium can even bioluminesce glow faintly in the dark. And just to show nature's complexity, it also forms a type of mycorrhizal relationship, an endotrophic one, with some orchids. So it's a pathogen and sometimes a partner.
Wow. Any other notable pathogens?
Crinopellis perniciosa causes a devastating disease in cocoa plants called witch's broom disease, leading to massive crop losses, particularly in South America. It illustrates a different type of parasitic relationship where the initial infecting phase, monokaryotic, is biotrophic, living with the host tissue, but the later phase, dikaryotic, becomes necrotrophic, killing the tissue.
Okay. And are there other, maybe less direct hazards?
Yes. Some edible fungi, like Macrolepiota, the parasol mushroom, can bioconcentrate heavy metals like mercury from contaminated soil to potentially alarming levels. So knowing where your wild mushrooms grow is important. Also, some wood-rotting fungi, like certain Phalaenus species, naturally produce chloromethane, which is actually a greenhouse gas that contributes to ozone destruction in the atmosphere.
Fungi affecting the ozone layer.
On a small scale, yes, but it shows their unexpected biochemical reach. And of course, as mentioned with Pleurotus, spores from many fungi, including common molds and wood rotters like Serpulolacrimans, can cause respiratory allergies or aggravate asthma in sensitive individuals exposed to high concentrations indoors.
Okay, now here's where it gets really interesting for me. Let's look at some of their amazing adaptations and behaviors. Have you ever seen a fairy ring in a lawn? Those perfect concentric rings, stimulated grass, then dead grass, then maybe more growth inside? It's almost like magic.
It feels like magic, but it's pure fungal ingenuity that's typically caused by the mycelium of a fungus like Marasmius oreades expanding outwards radially in the soil from a central starting point.
So it's a growing circle of fungus underground.
Exactly. The outer ring of stimulated greener grass is often where the actively growing edge of the mycelium is releasing nutrients by breaking down organic matter. The zone of dead or dying grass just inside that might be where the fungus has depleted nutrients or perhaps even produced toxins that affect the grass roots. It's a literal ecological footprint, a visible sign of the fungal colony claiming its territory underground.
I remember discovering my first fairy ring as a kid, and it felt like pure magic. It's fascinating to know it's a literal, well, ecological process happening beneath the grass.
It really is.
And speaking of ingenuity, let's revisit gravitropism. We mentioned it briefly with gill orientation, but can you give us another example of how fungi fight gravity?
Absolutely. Flammellina velutipes, the velvet shank or enoki mushroom, is a perfect laboratory example. If you take its growing stalk, or stipe, and lay it horizontally, it will literally bend upwards against gravity to reorient itself within hours.
How does it know which way is up?
The cellular mechanism is wild. It involves differential cell enlargement on the lower side of the bend. It's thought that the nuclei within the cells act as statoliths, dense particles that sediment downwards due to gravity, and this somehow triggers signaling pathways that influence cell wall growth and expansion to cause the upward bending.
Nuclei acting like little weights. That's amazing.
It is. While Flammellina is quite slow and bends mainly near the tip, another ink cap, Coprinus cinereus, responds much faster and its stipe curvature extends along the entire length. This probably reflects its very ephemeral, short-lived nature and needs to get upright and disperse spores quickly. And get this for sheer force. Coprinus turquolinus, another species, has been observed in experiments to lift weights of over 200 grams, nearly half a pound, with its elongating stipe.
No way, a mushroom lifting half a pound.
Yes. Imagine the force needed for a mushroom to crack through asphalt or lift paving slabs, which they sometimes do. It's generated by turgor pressure during cell elongation.
That's truly astounding. They are far more powerful than they appear. And what about competition? Do fungi actively fight each other down there?
They absolutely do. A fascinating behavior studied in detail is hyphal interference. Fungi, like Coprinus heptemeris, when their hyphae encounter the hyphae of a competing fungus, can defend their resources by literally killing the competitor's hyphae on contact.
Oh.
It involves a rapid loss of turgor, that internal water pressure that keeps cells rigid and seems to trigger an oxidative burst in the sensitive hyphae of the competitor, causing membrane damage and death. It's a direct, ruthless form of chemical warfare for resources, ensuring they dominate their patch of substrate.
So it's a constant microscopic battle for resources down there, and these fungi have evolved incredibly effective, sometimes brutal, ways to claim their territory.
It really is a microscopic jungle.
And finally, what about a fungus that can survive extreme conditions? Something really tough.
That brings us back to xeromorphic adaptation. Beautifully illustrated by Schizophyllum commune, the split-gill fungus. We find it everywhere. In dry weather, its unique split gills literally curl inwards along the split, rolling up and protecting the spore-producing hymenium inside.
Like closing up shop.
Exactly. Then, when moisture returns, even just dew, they uncurl and straighten out again, ready for spore dispersal. This fungus is incredibly resilient. It can even revive and produce spores after years of complete desiccation, being totally dried out.
Years. That's incredible survival.
It is. It's a remarkable survivor, and partly because it's so widespread and easy to grow, it's become a key model organism in labs for studying fundamental fungal biology. especially complex fungal mating systems. It's a real testament to their incredible adaptability.
What's fascinating here is how these organisms operate on such individual yet coordinated levels. I've seen those dark zone lines in decaying wood sometimes, dividing different patches of fungus. What's actually happening there?
Those zone lines are incredible visual evidence of fungal individuality in competition. In wood-decaying fungi like Tremites versicolor or Mysterium hirsutum, individual dikaryotic colonies, genetically distinct individuals, can persist for years within a single log. Where two incompatible colonies meet, they often form these visible zone lines of antagonism. These lines can be made of dense, dark hyphae, sometimes with altered wood structure. They're literally walled-off battlefronts within the wood, preventing the mycelia from merging.
So each patch is its own territory.
Exactly. And these individual colonies continually produce fruit bodies from their established territory. It showcases the complex society of a single fungal species within a larger substrate, constantly competing for space and resources. It raises an important question. How do these individual colonies maintain their genetic integrity and boundaries in such a competitive environment? It's a microscopic war zone with visible borders.
Amazing. Okay, to try and wrap this up, we've journeyed from just the superficial appearance of fungi to their intricate microscopic structures. We've explored their vital roles as decomposers and symbionts and uncovered their really profound impacts on our food, our medicine, and our environment, both beneficial and sometimes quite harmful.
Yeah, I think it's clear that Homobasidiomycetes are far more complex and influential than their common appearances might suggest. Their hidden mechanisms, their diverse life strategies, they're truly remarkable, constantly adapting and shaping the world around us in ways we're only just beginning to fully appreciate.
So what does this all mean for you listening? Well, next time you walk through a forest or even see a mushroom pop up in your garden, maybe take a moment. Remember the unseen world teeming beneath your feet. Remember the powerful decomposers recycling nutrients, the hidden networks connecting entire forests, the surprising compounds that offer both healing and danger. What other hidden wonders are quietly at work just beneath the surface, waiting to be discovered? Thank you for joining us on this deep dive into the fascinating world of Homobasidiomycetes. This has been a last-minute lecture special, and we really appreciate you tuning in.